Hello, everyone, and welcome. My name is Adrienne Messe with Organic Trade Association. I am very pleased to welcome you all to today's webinar brought to you by the Organic Center. Today, we're going to be learning about crop-livestock integration of organic produce crops, impacts on food safety and soil health. Today, when the webinar is over, you are going to see a survey pop up in your browser. We would really appreciate you taking the time to complete this pretty brief survey. The more responses that we get to this survey, the better, so that these researchers can use their data for their next steps with all this important information and research. Thank you so much in advance for taking just a couple minutes at the end of today's webinar to complete that survey. Today's webinar is being recorded. Any resources that we share today in the chat or our reference from our presenters will be shared to you by email within the next 2 business days. All registrants will receive a follow-up email that includes the PDF slides of the presentation, helpful links, and a registration link to view today's webinar afterwards if you'd like to rewatch it after today. Feel free to share all of this information out with your colleagues and your networks. The more that we can spread this information, the better. So once you receive that follow-up email, don't hesitate to share around with your network. During registration for today's webinar, we asked that you let our presenters know any questions that you have on the top of your mind relating to today's topic. We are very thankful to have a very diverse and engaged audience joining us today from around the world, and we really appreciate those questions that you submitted ahead of time. We've taken those questions into account for our panelists' presentations and in preparation for the Q&A session taking place later today in the webinar. And for any other questions that come up throughout today's discussion, please feel free to use that Q&A box on the Zoom platform you see at the bottom of your screen. Our presenters will respond to your questions as time permits during our 2 Q&A sessions, and we will address your questions as best we can or follow up afterwards. We're going to focus specifically on questions that are relevant to this particular work. If you have other questions for our presenters or the Organic Center, don't hesitate to reach out at any time and we can continue the discussion. So a little bit more about the Organic Center. The Organic Center is the trusted source for the science of organic farming and products. We convene credible, evidence-based science to fill knowledge gaps, support organic agricultural professionals. We work collectively with many stakeholders across the industry to advance organic by performing research, developing resources to promote the benefits of organic, and communicating the organic difference to consumers. The Organic Center is the sister science organization of the Organic Trade Association. Organic Trade Association is a membership-based business association for all organic agriculture and products in North America, whereas the Organic Center focuses specifically on the science behind organic. You can learn more about both organizations, including membership benefits that are brought to you through joining as an Organic Trade Association member in the links that I'll post in the chat in a minute, and you'll also receive that info in the follow-up email afterwards. So we are going to dive into today's webinar in just a second. So let me introduce our esteemed panel of speakers for today. We are so grateful that these individuals have taken time out of their busy research lives to present this info to you all today. First, we have Dr. Alda Pires, Associate Specialist in Cooperative Extension at UC Davis School of Veterinary Medicine. Also from UC Davis are Sejin Chung, PhD candidate in epidemiology, and Sequoia Williams, MS candidate in soils and biogeochemistry. From the University of Minnesota's College of Food, Agriculture, and Natural Resources, we are joined by researcher Emily Evans. And 2 speakers who are joining us today, but not formally presenting. They'll be joining us for the Q&A at the end to be able to answer any questions that come up in the chat that are relevant to their work. And that's Dr. Amelie Goudon, Associate Professor of Agroecology at UC Davis, and Fawzi Hashim, Research Associate Professor at the University of Maryland, Eastern Shores Department of Agriculture, Food and Resource Sciences at the School of Agriculture and Natural Sciences. Thank you so much to all of you for sharing your expertise with us today. I know I am definitely going to learn something throughout this webinar for sure. So now I will hand things over to my lovely colleague, Dr. Amber Sciligo, the Director of Science Programs for the Organic Center, to talk about today's presentation, our research partners, and a little bit more about our panelists. Thanks so much, Amber.
Thank you, Adrienne. I'm so happy that you have joined our webinar production team. So welcome. Hello to everyone else. It's exciting to have you all here for another Organic Center webinar, where today we are gathered here to talk about the wonderful world of food safety yet again. This is a theme that has gotten a lot of attention in the past, but it continues to stay popular as food safety regulations remain diligent. And then, of course, organic farmers are always continuing to search for the balance between meeting those necessarily strict regulations while also satisfying organic standards, and then also continuously improving the environmental impacts of their operations along with their economic feasibility. So finding that balance can be a challenge, and it is a tall order indeed. Before I dive too deeply into today's topic, though, I did want to share a little bit more about why the Organic Center is hosting this webinar. As Adrienne mentioned just a minute ago in her introduction, one of the main goals of the work that we do at the center is to help fill knowledge gaps across the supply chain. And we do that from the farm to the table. And one of the ways that we accomplish this is to facilitate research that addresses production challenges for organic farmers. And this wheel that you're seeing here shows you, this high level view of some of the different phases of that research facilitation process. Starting with identifying our knowledge gaps, which is the top right bluish. I'm not even going to know what that shape is, but anyways, sort of corner in a circle. So we start with identifying these knowledge gaps, and we do that by engaging with various stakeholders across the industry to see what kind of research is really needed. And then we help pull together research teams by reaching out to our network of academic researchers and encouraging them to develop projects that will meet these research needs that we've identified. Sometimes we will help them raise funds through private and public sources. One of the most important public sources of funding for this kind of work that we've been doing is the USDA-ARS program. And these days, they offer up to $3.5 million for a single multi-institutional project. So there's a lot of work that can be done with this funding, but we also access other funding sources too. But once we have that funding in hand, then the research can begin, and the Organic Center will fit in however the research team would like us to. Sometimes that means we're very involved down to the project management level. Sometimes that means that we are less involved and we sit in more as communications partners where we share their work with the public. And this webinar is an example of that public sharing. We also host a website for this project, which you can find. Maybe somebody from our team can pop that link into the chat for all of you. But you can find more work throughout the process of this project from them at that website. And then ultimately, we aim to leverage the research results to make a broader impact. And whether that's to offer more understanding of the benefits of organic to consumers, or to offer practical solutions for farmers, or to put science behind asks to Congress to improve policies that support the entire organic industry. So we are here today to share with you some results from this long-standing research program. And I'm really hoping that all of you walk away having learned something meaningful today. Next slide. Okay, so Dr. Pires is going to start us out with some background on this project, why it's important, why they chose to study different regions across the US, and give a general overview on the science behind this work. Then she will be followed by Sequoia and Emily, who will share what this work looks like in the field in these different study regions with their field demonstrations. Then we'll come back to Sequoia, who will present some results on soil health and nutrient cycling. And this will be followed by the implications for food safety with results that are presented by Sei-Chin. And finally, I will round us out with a quick reflection on how this work applies across the industry before moderating our Q&A session. Please feel free to ask questions throughout in the chat, and we'll get to them at the end of all these presentations. Next slide. All right, we're going to get some deeper background information on this project specifically. Oh, I think maybe we went too far on these slides. There we go. Perfect. But I wanted to take us way back to the beginning of this work, which began roughly 10 years ago, maybe not quite that long ago, when the FDA proposed revisions to the Food Safety Modernization Act. And those revisions included changes to the required wait period between the application of untreated manure and the harvest of the treated crop. At that time, the FDA proposed a 9-month minimum interval requirement, and this directly contradicted the National Organic Program regulations for the application of raw manure, basically creating an impossible situation for organic farmers. So that ruling was not adopted because FDA decided that further research was needed. And with that recommendation, the Organic Center reached out to Dr. Alda Pires and some other academics across the country to help develop research that was aimed at making sure that organic systems were included in the collection of additional data that the FDA would be considering for future rulemaking. Some of that research was presented across 3 webinars that we've hosted in the recent past couple years. And today's webinar is actually an extension of that work that began way back then, because the program has evolved with different areas of focus and different funding sources. Today, we will be discussing food safety impacts, specifically of cover crop grazing and fresh produce systems, with a focus on cover crop adoption, crop livestock integration, and soil health. Next slide. Okay, so we know that planting winter cover crops and integrating livestock into cropping systems are both practices that can have important benefits for growers and the environment. But these practices don't come without some challenges to pair with those benefits. So for instance, cover crops, we know, have a lot of benefits. They can enhance soil fertility, the soil structure, including water infiltration and water storage, and also help reduce nitrate leaching. However, there are still challenges in expanding cover crop use, partly because of concerns over the water that's needed to grow cover crops, and also nutrient immobilization, which basically means that some nutrients can get locked up in the soil and are harder for plants to uptake. And then this can lead to increased input costs for the following crop. These are just a couple of the concerns. Many growers do also consider livestock grazing of cover cropped fields in fresh produce operations as a way to enhance soil health and other environmental benefits, but there are also concerns about microbial food safety, which have been limiting the expansion of this practice. And that's because there's concern that livestock grazing can introduce fecal-borne, food-borne pathogens into the soil, and then there's a potential for transfer to harvested produce that will be consumed raw or minimally processed by consumers. Next slide. So to address some of these concerns, the goal of this project is to evaluate the food safety and soil health impacts of ruminant grazing of cover crops. And the team aimed to do this by measuring several things: food pathogen persistence and survival in soil, as well as transfer to vegetable crops, changes in soil health indicators over 2 years of grazed cover crop vegetable production, and the benefits and potential trade-offs for crop productivity. And then this experiment has been conducted across 3 states with different climate conditions and growing seasons to see whether the benefits and challenges are regionally dependent. Next slide. This project is led by the University of California in partnership also with the Organic Center, USDA's Agricultural Research Service, the University of Maryland Eastern Shore, and the University of Minnesota. And I'll mention also that the funding for this work specifically was made available by the USDA's Agricultural Marketing Service. Next slide. Right. Okay, here we are to the good stuff. I think that's plenty of talking from me. I'm going to go ahead and hand it over now to Dr. Alda Pires to give you all more detail about this research project. Thanks.
Good morning or good afternoon. Thank you, Amber, for the introduction. So I will just, take some time to next slide. I'll start with just overview. Amber did a great introduction about overall and the reasoning for this project. Just overview for what we consider integrated crop livestock systems. Also, sometimes you see in the literature diversified farms. So they're farms that animals and crops are raised with utilizing the products of one growth to the other. And here we have pictures of some of those types of integrated crop livestock farms. On the left, you have use of poultry. So they have these egg mobiles where poultry are moved along of semi-permanent pastures. And another livestock or animals would use frequently are sheep. They might be used in residue crops. So after that, those crops are harvested for human consumption. So here is on sweet corn or cover crops, and that's the specific aim of study of this presentation. There are many benefits, as Amber mentioned before, of this rotational grazing, so moving animals from pasture to produce areas, either cover crops or residue crops, and as many benefits in terms of soil fertility and organic matter. But one of the challenges we're using manure, and most of our animals, livestock, are reservoirs for foodborne pathogens of public health concern as Salmonella, E. coli, O157, and Listeria. So how we consider this is identical, the risks in terms of food safety manure application. And so that implies a lot of planning, one to integrate these animals in the systems on the production of vegetables of what we consider high risk for contamination. And so what the growers are doing are actually, they follow the organic certified growers on fresh produce. They follow USDA National Organic Program of 90 and 120 days. So that's between the period of the grazing and when those crops for human consumption are harvested. And as Amber mentioned, this was some discussion before when FSMA released the PSR, so produce safety rule, in terms of applications of manure. What happens with grazing is compared to untreated manure, we know there is less deposit of manure in terms of the rate and during rotational grazing. And in FSMA, actually, the introduction of animals, they are in the same part as wildlife intrusion. Could be that considered as a wildlife intrusion. However, for the farm perspective, easier if we have a time interval. Oops, okay, so next slide. So what we know, there's limited research to assess these risks on food safety. What are the risks of integration of sheep on these cover crops and fresh produce type of production? We did have a work done by our team that looking at on farm some of those risks, although it was very limited in terms of study was focused just on one farm, and we didn't assess foodborne pathogens specific. So what do we know about the risks, potential risks of introduction of animals on other crops? We know that the foodborne pathogens presence in animals or the prevalence in animals, it depends many different risk factors, health status of the flock, the prevalence for Shiga toxin, E. coli, one of the foodborne pathogens of concern, it might range from 3 to 30%. They also can carry other foodborne pathogens as Salmonella. The diet might matters. Also, if they are submit animals to stress events as transportation, heat stress, density, livestock density, it may affect the shedding of these pathogens. Normally, these animals are healthy, so you don't see they are sick, and they can carry these pathogens in their GI and shed intermittently. Once these pathogens are in the soil, there are studies on foodborne, on manure and other soil amendments have shown there's another factors may affect that ability of that pathogen to survive in the soil, to the environmental factors, inclusive weather, specific to the soil characteristics as soil type, micropopulation, and so on. The cover crop type also, there is some evidence they might change that micropopulation in the soil. So with that, we got funding for initial start as actually a California project, and then we expand to another states. And the main objectives of these, the three main objectives of this multi-year project, we finish up the third year, was assess of the cover crop systems and sheep grazing on foodborne pathogens. And because those pathogens are rare, we use also indicators of contamination to monitor that potential contamination of fresh produce. Also looking at nutrient cycling, soil health, and yield as well, and then having the outreach program that will share some of these learning or the findings and some of the recommendations of these systems. This was conduct, as opposed to our initial work was conduct on a research farm here in California, Russell Ranch, in Minnesota, in Southwest Research, and also in University of Maryland. So these were research farms where either we use our sheep or we bring external animals to these plots. They were all certified organic for this specific field trial. So the study design, it's the same for all three states. And here just overview of the study design. I repeat this study 2 years to capture 2 growing seasons, and was randomized complete block study design. So you have 4 blocks where we assign 3 treatments to follow no cover crop that was seeded normally in the fall. And then one of those plots were grazed and another was not grazed. So this is the same layout, and just give an idea in the next slide where we have, this was an example of California, you have the graze cover crop where we move sheep from one plot to another plot, and then the no graze was the cover crop was terminated and tilling, and they have another treatment was followed. The sampling scheme will change depending what was our outcomes of interest, and you will see this slide repeated in the next slides presented by Skoya and Sajin, but just want to give an overview. So if you look at this is the timeline for the 2 years, so we repeat the study 2 years. We see the winter cover crop in November or December for the case of California, and other states was different, as you're going to see in the next presentation. And then we brought sheep or goats to the cover crop when it was mature to graze between 3 to 7 days. For the soil-borne pathogens, we sample those plots before the grazing, after grazing, and every 30 days to see if we could isolate or not the foodborne pathogens. And for the soil health outcomes was twice a year. The first year we did plant, we did transplant spinach, and then the second year used cucumber. And the idea here is to use 2 different high-risk crops that potential contamination can be cross-contaminate. They're very close contact with the soil. And also 2 crops it could grow across 3 states. So we have a very different growing seasons in California compared to South Maryland. That's why the reasoning for this. With that, so Skoya and will present next presentation. She will give overview what looks like the field trials of our first and second year and some of the challenge through the field work for this project.
Great, thank you, Alda. Hi, everybody, I'm Sequoia. I'm a master's student at UC Davis, and I helped run this experiment at Russell Ranch. Since we can't be in the field with you all today, I'm just gonna show you some pictures from our experiment so you can get an idea of on the ground what our sheep integration trial looked like and what issues we dealt with. So first, to set the stage for you all, we're in Winters, California, and this is a Mediterranean climate. This is showing our average temperature and average rainfall in this area. We don't get snow like Maryland and Minnesota, so we have a much wider growing window. It's about from mid-February to mid-November, and so cover crops are great because we plant them in November, and then they kind of take advantage of that winter rain that would otherwise maybe not get utilized. And then this is kind of another diagram showing our cropping sequence and grazing events. So we seeded our cover crop mix in November of both years. It was a mix of cereal rye, crimson clover, and daikon radish. Then we had grazing events, one grazing event each year. And then the first year, like Alda said, we had spinach. And then the second year we grew cucumbers. Okay, so now here are some of the photos from our field trial and some videos too. So this is showing we kind of seeded the cover crop in November of 2020. We actually didn't have a germinating rain until about a month later, which is a common thing that growers in California have to deal with, is trying to get their seeds in the ground before the first rain, which is always uncertain when that's going to be. So the seeds kind of sat there for a while. It was a very dry year, but by February, we had some good cover crop growth. Even though we planted that mix of 3 different species, it was heavily dominated by cereal rye that year, which was interesting. And then finally, by about March, there was enough cover crop biomass to graze it. We had about 1,500 pounds of dry biomass per acre, which was quite a bit actually. And we used about 50 sheep for 2 days, grazing our 4 different blocks. We had to have like night watch of our sheep because there was no livestock guardian dogs. So we had to kind of keep them safe that way, which was a little bit of a challenge. It was quite tiring. And yeah, so we had that one cover crop grazing event this first year. Then in spring, so shortly after the grazing, we terminated both the ungrazed and the grazed cover crop. And then you can see this video is going. It's the video showing how we planted the spinach. So we grew some spinach in the greenhouses, and then we transplanted it using this paper pot transplant system. Then we surface irrigated our spinach with the surface irrigation system, and that helps keep the spinach cool, and which was important in California because we were worried about it bolting really quickly because it's so hot here, even in springtime. But we did have some weed pressure with this system because we were sprinkling the surface, so there was a lot of weed competition with the spinach, which is a common concern that organic farmers have to deal with. So we had to have a crew weed the field for us, hand weed the field for us. And then about 6 weeks after we planted, spinach grows really fast. So we already had enough spinach. We harvested the spinach after about 6 weeks in May. And we harvested the center bed of all of our plots to get yield estimates of spinach. And then we tried to see if it would regrow and if we could get a second harvest, which is common with spinach, but it was too hot and the spinach bolted and we had a lot more weed pressure. So we didn't get a second harvest off of it. So then after looking at the second year, after spinach, the field kind of sat for the rest of the summer, and then we seeded the cover crop mix again in November. This year was a little bit more moist. We actually had rain before we seeded the cover crop. So the field was kind of already saturated, and we had a totally different mix that popped up this year. Rather than the first year, we had a mix dominated by daikon radish, even though we had seeded the same exact mix. So it kind of shows how years are very variable when it comes to what grows with cover crops. And since we had more moisture, the cover crop was growing a little bit faster. We actually could graze it a little bit earlier this year. We grazed it in mid-February. And we had about 25 sheep graze it for about 1 day. So we were moving them kind of quickly over the field. they hadn't really grazed daikon, I think, before, so they weren't super interested in eating the daikon. They would nibble on it, but they also were into eating like the weeds that had popped up as well. And they did a lot of trampling of the cover crop as well as grazing it. And then this video should show kind of how we moved the, let's see, I don't know if I can play it. Yeah, so this is just kind of showing our elaborate fencing that was required to move the sheep from one grazed plot to another, making sure they didn't accidentally graze the ungrazed cover crop, which was a success. So that was good. And they always love moving to the fresh cover crop patch. And we're very excited about it, even if it was just daikon. Okay, so yeah, after that grazing event, we terminated both cover crop treatments, both ungrazed and grazed, and then we direct seeded cucumbers into the field for this year. So we had to pre-irrigate a little bit before we direct seeded, and that might have been a mistake because we ended up having a lot of cucumber beetle damage on our that prevented emergence of our cucumbers, and we also had some frost damage. So there wasn't a ton of success in terms of the cucumbers, but we still managed to harvest some for yield estimates or for foodborne pathogen analysis. We also had to do some hand weeding to keep the weeds under control again. And then, like I said, we did get some samples. Sejin got some samples for foodborne pathogen analysis, but we didn't get any samples for yield estimates. So that was the trial in California. We learned a lot about how the struggles of organic farming and timing and implementation of grazing on top of those challenges that organic farmers face. And now I'll pass it over to Emily Evans, who will talk about how the trial went in Minnesota.
Thanks, Koya. I'm Emily Evans, and I'm a research agronomist at the Southwest Research and Outreach Center in Lamberton. I, along with Lee Klossner and Dr. Paolo Pagliari, manage the Minnesota site, and that's located in the southwest corner of the state where that little yellow star is at one of our research centers. The area of the experiment was certified organic since 2008, and we grazed goats rather than sheep at our site just because that's what we had access to. Next slide, please. So this is our typical growing season. You can't really call it typical because it seems like it changes all the time, but it's considered May through September. You can see the 30-year historic averages above and below the bar show how there's the milder temperatures during that time allow for crop growth. But it has to be said that we also get 70-degree weather in March and sometimes in October and November. And then there are times in May that we get blizzards that yield a foot of snow. So we have very variable weather. This short growing season makes it a little tricky for cover crop establishment, especially, but it's typically thought that if you can plant a cover crop by mid-September, you'll get enough growth to make it agronomically worthwhile and to have protected soil. Also, the harsh winter that we have here kills many cover crops, but sometimes this is actually considered helpful because we have such cold, wet springs here, and that's because of massive snow melt and heavy spring rains. So sometimes that physical termination of cover crops is really difficult. So if you take that task out of the equation, it can allow for more timely planting of those main crops. Next slide, please. So this is the cropping sequence in each of the 3 years of the study. We grew the same cover crop. It's the tillage radish, winter rye, and berseem clover. All three crops typically establish well in the fall. However, the clover and the radish crop will both winter kill with the cold temperatures. And then the winter rye just goes dormant and then regrows as soon as the soil temps warm up. So in our situation, we could actually have two grazing events for each cover crop planting. So in the fall of the first year and the fall of the second year, the goats grazed on all 3 species. And then the spring of the second year and the spring of the third year, they just grazed on the winter rye regrowth. Also, as I said before, in this area, planting between August 15th and September 15th is ideal for good establishment. And both years we got it end of August and early September. So following that first planting on August 27th of that first year, we had great rains, warm fall temps. Whoops, we skipped ahead. We can go back one slide. Yep. Then right before grazing, we got 6.5 inches of snow within a week, and the temps dropped down to 2 degrees Fahrenheit. So this is before we had the goats come and graze. So this did some damage to the tillage radish. You can see that in the picture on the far right. Yet still right before grazing, we had just a little less than 1,400 pounds of dry biomass of cover crop per acre. Next slide, please. So then when the goats came, that day was actually 79 degrees. So that was within a few days of that 2 degrees Fahrenheit low. We had a high of 79. We had a herd of 40 goats that was brought by a goat dispatch, and which is basically a rent-a-goat business. And they dropped it off and fenced it in, very similar to what California had. And they stayed for one very long day and grazed on each plot for approximately 2 hours each. Next slide, please. And this is just another example of the wide range and drastic change in weather. So after that 79 degrees grazing day, we had a week of those beautiful temperatures. And then a week after that, we were sampling in another 4 inches of snow. So that's kind of how our falls and spring go. Next slide, please. So this is the following spring, and in March, then we had unusually warm temperatures, and this really kickstarted that rye regrowth. And in general, that winter was considered pretty mild. Next slide. And because of that mild winter, we did have some berseem clover grow as well as the rye. So you can see that in that center picture. And prior to grazing, we had about 1,200 pounds of dry biomass per acre. And the herd that came this year consisted of 115 goats grazing over 3 days. And this was my favorite grazing event because we actually witnessed 2 births, yielding 4 babies, and they were born in our plots. So we had 1 single birth and a set of triplets. So that was kind of fun. Next slide, please. So this was our first main crop that summer. After spring grazing, we terminated immediately the winter rye by mowing and then disking the field a few times to ensure that we had an adequate kill of that winter rye and also to prepare a decent seed bed. And our first main crop, just like California, was spinach. It was a 5 by 20 foot plot. We had smaller plots just because we didn't have enough manpower to manage larger plots. We planted the spinach from seed May 10th, and we had such heat and super dry conditions. We couldn't really keep up with it, and it was really difficult to get a good establishment. So we did replant June 8th. The crop just still did not grow well. It was super dry, super hot. So per plot, we had just about 1 pound fresh weight. Next slide, please. So for the fall planting of the cover in year 2, we prepared the soil August 23rd, just by disking the area a few times to kill any existing weeds, and also, again, to prepare the seed bed. We planted the cover crop September 2nd, and in just a little over a month, we had a beautiful cover crop that was ready to graze. Next slide, please. And so right before our 3rd grazing event, we had close to 900 pounds per acre of dry biomass. And so for this event, we had 160 goats that grazed the 4 plots. And then after the herd left, just like we did the year before, I hadn't mentioned it, whatever remained of the cover crop was left to act as a soil cover for winter protection. Next slide, please. And then in the spring, it was interesting. We observed a trend of the rye regrowth, where the grazed plots produced more than twice the amount of rye biomass. So the plots that were grazed produced over 1,400 pounds of dry biomass compared to the ones that weren't grazed, which were just a bit over 600. So this likely is because the goats grazed mostly on that tillage radish in the fall. And so this, obviously left room for better establishment and fall growth of the winter rye for that month, because they grazed in October and they still had a couple weeks of, we still have a couple of weeks of good weather. I think we didn't see this the year before because the goats didn't really eat the radish in the fall the year before because we had that frost damage. So that may be why we saw what we saw. Next slide, please. And so our fourth and final grazing took place on May 20th. We had a herd of 170 goats and they grazed for a full day, spending about 2 hours in each plot. And you can see where the rye is super tall here. Next slide, please. And so that brings us to our main crop that third year, which was cucumbers. Immediately after the goats left, we mowed and then disked again to ensure complete termination. We transplanted cucumbers on June 2nd, and all plots yielded quite a bit of fruit. The average was over 80 pounds of produce per plot, and the plots were 5 foot by 20 foot. Another interesting observation in this third year is we saw that the fallow plots yielded double compared to those that were planted with a cover crop, even though all plots were managed and watered equally once they were planted to cucumbers. And it's probably because those fallow plots started with a higher soil water content at cucumber planting compared to the plots that were planted with cover crops, because the cover crops were taking up water throughout the spring. So that water competition, we really saw it with this cucumber crop. Next slide, please. And that's the end of the study at the Minnesota site. So I had to leave with this handsome fellow. I kind of fell in love with the goats the last few years with them at the Minnesota site. But now I'll turn things over to Sequoia again.
Great, thank you, Emily. Okay, so I'm back again. I'm going to talk about soil health now and the soil health outcomes of our trial at Russell Ranch in California. So first, what is soil health? It is a term that is thrown around a lot right now, especially in the agricultural space. And so a healthy soil is anything that's a soil that's alive and is that providing functions to sustain plants, animals, and people. And another definition of soil health is the degree to which dynamic properties have been managed for optimum functions within the constraints of the soil's inherent properties. So these dynamic properties are properties that can change pretty quickly in the soil. And indicators of soil health and of these dynamic properties can be broken down into 3 different categories. There's physical, chemical, and biological indicators. So some examples of these different indicators. One physical indicator of soil health is compaction, which is measured with bulk density. Aggregate stability is a measure of aggregation, which is important for water infiltration into the soil and to prevent erosion. In terms of biological indicators of soil health, we have potentially mineralizable nitrogen, which is an indicator that measures how much organic nitrogen can be mineralized into inorganic nitrogen for crop uptake during the cropping season. There's also active carbon, which is carbon that's available for microbes to use in the soil. And finally, some chemical indicators of soil health are nitrate nitrogen, which is a snapshot of what nitrogen is available in one moment for plants, and pH, which is a crucial variable for all nutrient availability. So now when we think about the second part of the definition, within the constraints of the soil's inherent properties, so because I'm in soil science, I had to put this picture of all the different soil orders. So there's a lot of different types in which soil health means something different. And the potential for a soil to change the pH or increase organic matter is limited by the type of soil that you're working with. So in California, we have, my experiment was on the YOLO soil series, which is actually this picture on the left. That's the YOLO series. It's a young soil. It has moderate potential for organic matter accumulation. So when we think about the results of this experiment across 3 different states and 3 different climates and 3 different soil types, we're going to see potentially different outcomes because we're doing it on different soil with different potential. And then finally, now that thinking about adding animals into the equation and how animals are going to impact soil health, first thing animals do is they trample the soil, which could lead to soil compaction, a physical indicator of soil health. And they also consume this cover crop biomass and they convert it into manure and urine, which is higher in nitrogen concentration than the original biomass. And kind of like what Amber was saying about cover crops having potential to immobilize nutrients, animals do the opposite, where they kind of mineralize those nutrients and make them more available. So this diagram is showing those manure and urine inputs that I just mentioned that are coming into the soil as a result of grazing. There's also some plant responses due to grazing that could be happening, like increases in root exudates and root sloughing. And these components are feeding into the organic fraction of the soil. So we have organic carbon and organic nitrogen. Those are the food source for microbes to grow. And then these microbes are going to be feeding into soil organic matter. And then in addition to this organic side of the soil, we also have some inorganic nitrogen that can be converted from manure and urine and become plant available for uptake, or potentially lost from the system as through leaching. So given that diagram and our interest in understanding soil health with grazing, we asked 4 different questions. We asked whether grazing cover crop increases organic and inorganic nitrogen pools, whether it alters soil microbial processes, alters soil chemical and physical properties, and whether grazing can lead to greater stabilization of soil organic matter. So just as a refresher, we had 3 different treatments. In the winter, we had fallow, we had ungrazed cover crop, and we had grazed cover crop. And then in all of the plots, we were growing spinach and cucumber in the summer. And actually in California, this was a continuation of a previous experiment. And so this was year 3 and 4. We had maize and tomato the first 2 years, and then we added this on and we grew spinach and cucumber. And we sampled, like Alda mentioned, we sampled for soil health 2 times at crop planting and at peak nutrient uptake. So first, to kind of look at some of the yield outcomes of our trial, like I said, we did spinach yield estimates, and we found that there was greater yield in the grazed and ungrazed cover crop plots. For spinach, for cucumber, like I said, we didn't get yield estimates that year. And now getting into some of the soil results that might help explain some of these differences in yields that we saw. First of all, for soil physical health, we didn't find any difference in aggregation or compaction with grazing, which is promising since that is a concern with a lot of farmers. For soil chemical properties, we did a whole suite of a lot of different soil chemicals in the soil, the whole nutrient panel. We didn't see any effect on soil salinity. We did see that ungrazed cover crop and grazed cover crop both decreased the pH of the soil from about 7.5 to 7.2. And there was also a slight increase in the availability of micronutrients in the grazed plots as well, although it wasn't of plant nutritional significance. And then as a final assessment at the end of these 4 years of grazing, we looked at total carbon and total nitrogen in the soil, and we looked at it at 3 depths from 0 to 15, 15 to 30, and 30 to 60. Fallow is in gold, ungrazed cover crop is in green, grazed cover crop is in blue, and you'll see that for the rest of my result slides. And we didn't see any difference between treatments in total carbon and total nitrogen. We did see like a decrease in the pools with depth, which was to be expected. However, when we were looking at more of the labile forms of nitrogen, we did start to detect shifts in the nitrogen cycle. So for potentially mineralizable nitrogen, which is the estimate of the nitrogen that's going to be converted from organic to inorganic forms for plant uptake during the growing season, we did see that grazed cover crop had the highest. And you'll see this trend in some of my results as well, like fallow was lowest, grazed cover crop had the highest, and ungrazed cover crop was in the middle. And similar to the potentially mineralizable story, we also saw that there was higher nitrate in the grazed cover crop and the ungrazed cover crop plots at peak nutrient uptake during the crop. But when we're thinking about this pool of nitrogen, we're not just interested in a snapshot of nitrogen, we're interested in this timing of release from organic pools into inorganic pools. So I looked at a ratio of the organic nitrogen at crop planting to inorganic nitrogen at a different time point at peak nutrient uptake. And I found that the grazed plots had a ratio closest to 1. So they were showing that the grazed plots had kind of optimized this timing of release of organic nitrogen into inorganic forms for plant uptake. And then we're thinking about all this nitrogen being released in the soil, but so it's the microbes that are kind of doing this release, and they're doing that through their activity and through their respiration. So microbes can respire carbon, CO2, just like we can. So we looked at respirable carbon, and we saw there was a trend towards greater respirable carbon with the grazed plots. However, this was not reflected in microbial biomass carbon, so there was more activity in the soil grazing, but it potentially was coming from the same size pool of microbes in the soil. And so these microbes in the soil are what are driving this microbial carbon pump. They're consuming labile carbon in the soil, and when they die, their necromass can be stuck to mineral surfaces, and that creates a pool of mineral-associated organic matter, which is a portion of the soil that is more stable carbon. So I looked at these different pools in the soil. Essentially, we have the mineral-associated organic matter pool, and we have the particulate organic matter pool. The particulate organic matter pool is kind of the beginning of this microbial carbon pump. It's the pieces of plants. It's like undecomposed material. And then this mineral-associated organic matter is organic matter that's been through microbes. The microbes have died, and now they're stuck to minerals, and it's going to stick around for longer. So I looked at those two different pools in the soil after 4 years, and we didn't see any changes in those pools with grazing. Although, like I said, we see shifts in the microbial community. The activity of the community, it didn't reflect yet in these like longer cycling pools of carbon. So some takeaways and next steps. In general, we saw that there was a large cover crop effect. So ungrazed cover crop and cover crop tended to have increased nitrogen, but grazing improved the timing of organic nitrogen release into inorganic pools for crop uptake. There was no shifts in total soil carbon or nitrogen, or in the proportion of carbon in mineral-associated organic matter or particulate organic matter. And then we didn't see any alterations with compaction. And then in the future, we're going to investigate more about how these outcomes vary across our 3 states and our 3 different soil types, and work on creating a more complete nitrogen budget of grazing in cover crops. And so with that, I'm going to hand it over to Sejin, who's going to talk about the concerns about foodborne pathogens.
Thank you, Sequoia. Hello, everyone. My name is Sejin Chung. I am a PhD student in epidemiology at UC Davis in Dr. Pires lab. And today I'll be presenting foodborne pathogens and weather risk factors in this organic integrated crop livestock system from our 2-year experimental design in California and Minnesota. So for the food safety aspect, we had 2 objectives. First, we investigated the potential soil and produce contaminations by foodborne pathogens in this system and compared them with non-grazed fields. And the foodborne pathogens we tested for were E. coli 0157, non-0157 staph, and Listeria monocytogenes, because these pathogens were the main causes of the foodborne outbreaks in the US. And secondly, we tested for the persistence of generic E. coli in the soil by sampling soils over time. And generic E. coli here has been used as an indicator of fecal contamination and a surrogate of SDAG in the soil. And additionally, we investigated the associations of generic E. coli and the weather risk factors. So as Dr. Pires and Sequoia explained it before, we had 3 different treatments in the experimental field to see the cover crop and grazing effect. The 3 treatments were fallow, non-grazed CC, and grazed CC. CC here stands for cover crops. So fallow had no cover crop and no grazing event. Non-grazed CC had cover crop but no grazing event. But grazed CC had both cover crop and grazing event. And as you see this timeline before, this timeline shows how we collected the fecal, soil, and produce samples each year. To test the foodborne pathogens in the field, we collected pre- and post-fecal samples, and soil samples were collected on day 0, day 7, and monthly after the grazing event until day 120 or day 150 post-grazing. And here the day 0 soil samplings were collected right before the grazing event. And produce samples, which are spinach and cucumber, were collected on both day 90 and day 120 after the grazing event. But there are some differences depending on the region. As you can see here, California used sheep, but Minnesota used goats for the grazing event. And in California, soil samplings were mostly done between February and July, but in Minnesota, those were mostly done between May and November. And this was due to the differences in timing of cover crop growth and the weather. Aside from the samplings, since environmental factors might affect the persistence of pathogens in the soil, we also retrieved some meteorological data from the nearest weather station from our experimental fields in California and Minnesota. And these are the factors that are commonly recorded in both weather stations. So after running univariable analysis to see the associations between these factors and presence of generic E. coli in the soil, the significance factors are included in the final model with the treatment effect and sampling times to find the risk factors. So this is the result of foodborne pathogens found in the post-grazing fecal samples remain in the fields. Unlike California, Minnesota had 2 grazing events each year due to the long winter. So the total number of samples collected were more than California. And for a little bit about background, SDAG can be harbored within the gastrointestinal tract of sheep and goats. So these small ruminants have been identified as asymptomatic reservoirs of SDAG. So we found 33 and 3% of non-015 SDAG positive from the sheep fecal samples in each year from California, and 42 and 65% of non-015 SDAG positive from the goat fecal samples in each year from Minnesota. And in Minnesota, we also found 30% of Listeria monocytogenes positive in the second year of the experiment. These results is the foodborne pathogens found in the soil samples. In the first year of the experiment, we did not see any foodborne pathogens in both California and Minnesota. And in year 2, we did not detect any E. coli 0157 or Listeria, but found some non-0157 stag in the soil from both California and Minnesota. This is the table when we got that stag positive samples by sampling days. All the stag positive samples were from non-grade CC or grade CC treatment, but not found any from the fallow treatment. Red colored value indicate that some of the stag positive samples were from grade CC treatment, and black numbers were from non-grade CC treatment. And we also found some stag in day 120 and day 150 post-grazing as well. Lastly, in the produce samples we collected, we did not find any foodborne pathogens in year 1 from spinach, but in year 2, we observed some non-0157 stag from Minnesota, cucumber harvested at day 120 post-grazing from grade CC treatment. And when testing for generic E. coli, 6 and 4% of California samples and 38 and 83% of Minnesota samples were generic E. coli positive in each year. However, when comparing three different treatments, there was no differences in the number of generic E. coli positive among 3 treatments in both states. And when seeing the persistence of generic E. coli in the soil by the proportions of generic E. coli positive by sampling day and treatment, the left side is year 1. In year 1, the proportions of generic E. coli positive were the highest on day 30 in graze treatment in both states. And in right side is the year 2. In year 2, it was the highest on day 60 in graze treatment in California, but on day 7 in graze treatment in Minnesota. So when pulling these all result into the modeling with the meteorological risk factors, this is the final table we got. The table shows only the significant factors among the variables we considered. So grade CC treatment has the significantly higher odds of being contaminated with generic E. coli than the fallow treatment until day 60 post-grazing, but no differences were found after day 90 among three different treatments. And the sum of 29 days precipitation before sampling days significantly increased the odds of having generic E. coli in the soil, but the average 2-day soil temperature before sampling significantly decreased the odds of having generic E. coli in the soil. So in conclusion, the 2-year data of this system show minimal risk of transferring foodborne pathogens from soil to produce after day 90 post-grazing. But when considering meteorological data, the application of USDA National Organic Program 90 to 120-day wait period between manuring and harvesting in this system should consider recent meteorological events. And for future studies, we need to include more other environmental risk factors such as water sources or presence of wildlife around the field to apply this result to other areas in the US. This is all I have for the summaries of food safety aspect for this 2-year experiment. And now I will hand this over to Amber.
Thank you, Sejin. Thank you also to all of our speakers, not just for their presentations, but also for tackling these questions as they've been coming in. We're going to move to a more formal Q&A session very shortly. So go ahead and pop your questions into the Q&A box. I know I said the chat box earlier, but I was wrong. I forgot that we have a different kind of platform with Zoom that's more evolved. So please, in the Q&A box. But to give you a little bit of time to put your questions in there, I just wanted to do a quick reflection on the results of this work. I'll say the thing that has been the most intriguing and fun for me being involved in this research team's work as it has changed over time and built over time is the differences that have that exist in the different regions and have posed different challenges for each individual research team, and then also like at an overall coordination level. So what I have seen is this really successful multi-institutional, long-term, multi-interdisciplinary project. And we can see that we need this work for organic systems because they're systems, and there are always, we can always think of more things that we should have studied. I've seen some great questions coming through. There was one that asked about higher levels of biodiversity measurements. That's another thing that could have been included in this. And there are so many more things too, but there are also so many challenges at each of these sites. And in this time that we have in our Q&A, what I would like to do is dive in a little bit more deeply into some of those nuances, because what I hear overall is that there are benefits to soil health and it's nuanced. There are minimal risks to food safety and it's nuanced. And so I'd love to touch base a little bit on some of those nuances. And then I'd also like to hear a little bit more from the team leaders in how it was to actually coordinate all of this work with all of these differences in climate, cropping systems, etc. So I think we've got a good number of questions that we can move into. So I'd like to invite the speakers back for a Q&A. And I don't want to forget to reintroduce two panelists that we have joining us who we did not hear from formally first as speakers. So we have Dr. Amelie Goudan from UC Davis, and we have Dr. Fazi Hashim from the University of Maryland Eastern Shore. So I'd like to welcome all of you back and these two new additions. And I think that I would like to actually start this Q&A. Probably this question to Alda, but anybody else who wants to join in. We hear a lot of interest in poultry. There's a lot of people who are interested in integrating poultry. And I know that you all looked at two different types of animals, but they were both ruminants. And my question to you is, well, it's two-part. One, are these results transferable? So if people are watching this and they're thinking about integrating poultry, can they look at your results and transfer them over to their system, or are there differences that need to be considered? And then two, are you planning on following up with poultry, or are you working with other partners, or do you know of other people who might be?
I can start with answer in terms of the food safety and what I've seen here, at least in California, the transition of integration of animals on these farms and majority like fresh produce and another type of crops. So when we talk with farmers, we'll see mostly poultry will be number one animal. They will be, as diversify their farm, or as extra revenue for laying eggs or for broilers. So there is a lot of interest of integration in poultry. We'll see most of the poultry, at least the ones I've seen, the poultry are kept in semi-permanent pastures, as I show. So they might have like 2-3 years on that semi-permanent pasture, and then they will rotate with other crops. So that's something integration we have seen. If they add, so I've seen some farms, they try kind of side by side. So two things to think about, they're totally different species, right? Birds and ruminants. So in terms of the cover crops and what are their nutritional demands will be very different. In terms of handling the animals, of management, having on labor for the farm, actually it's very different having a bigger animal than poultry, than a smaller animal. In terms of food safety, both of them can carry these foodborne pathogens in their GI, although poultry is well known for salmonella, right? The risks, number one is salmonella, and that's what is most of studies done on the poultry-related soil amendments has been on salmonella. They can carry E. coli on 5.7 STAC. It's less study, we have findings on it. So for our study, we're looking at E. coli on 5.7 STAC and Listeria because ruminants are reservoirs for Shiga toxin E. coli. So is that transferable? Will be interesting, another question, because they are different behaviors in terms of, grazing and different risks, and then Emily can talk about, the different inputs. I think they pose different risks that we need to look at. I know that is for the audience, there is a webinar coming up, and then we can share for a colleague here in collaboration with Iowa and Kentucky, specific for integration of poultry in this type of system. So if you're interested really specific for poultry, there'll be a webinar to attend on Friday, on 28th, we can share. That's what I have.
That's great.
My opinion.
Did anybody else want to add to that from another soil health or other perspective? No? Okay, great, thanks. Well, I actually was just thinking, your answer just reminded me of one of the results that Sequoia presented on some of the soil health metrics, and compaction was one that was brought up, and it was interesting for me to see that there wasn't much of a difference in the compaction, because this seems to be a concern for a lot of folks who are interested in integrating livestock. So that's not a question, sorry, that's a comment, a personal comment. But I did want to dive in a little bit more with Se-Jin about some of the questions that, some of the results that you had around weather, and your final conclusion that the NOP should consider meteorological events in their recommendations. So when I think about the implications of this work and providing scientific evidence for these policies that currently exist and changes that we potentially could ask for, how exactly do you see this translating to that policy? Are you saying that what we have is good and we need to consider something else, or should we be making some changes?
Since for the data analysis, we just consider 2 growing seasons in 2 different states. I think we need more broad in different areas, more data to confirm that if the NOP rules is surely can apply to everywhere. So as far as what I have now, I think we should consider more data about various regions to confirm that.
So I might add to that. We're still doing some analysis to look at those isolates. Are they coming from the sheep, or are they coming from something else? So she showed data that in Minnesota found isolates in cucumbers and also in the soil. And that's the importance to do this multi-year. Actually, for California, we sampled the same plots, the same system, different crops for 40 years. The 4th year, that's when I actually found pathogens after those 9 to 120 days. So what we see, compare this data with other applications of manure we have had in projects, much lower concentration. The question is, since the risk is less, but are those pathogens coming up from those from sheep or what other animals? So we follow pathogens in another study didn't show on non-grazed plots. So heading to that, we have a question about mulch in cucumbers treatment. We did not consider mulch for these settings. We see that farmers use mulch for different reasons, for weed control and so on. We did do on-farm project actually on tomatoes. They use mulch, and and we did collect the soil through the same timeline, and we found some of those pathogens at 90 days too. But again, what's the limitations of that work? Can we point to the sheep? We didn't have a resource for it. So in summary, what's the challenge of these rules is statewide, nationwide, and we see that's the big effects on these survival are environmental, so the soil, moisture, organic matter, and weather, and that's very specific of where you are in the country and what the weather brings to you. So yeah, so more thought, considered about these guidelines and how to minimize the risk. There will not risk zero, but how to minimize that risk.
Yeah, that's great. And to follow up on that, I'd like to ask Amelie to, from an agroecology perspective, when you're talking about integrating livestock, and now we are incorporating these elements of soil health and these risks of food safety, these social elements, economic as well as environmental. How do you talk to farmers? How do you about these results, and what kind of recommendations do you make? Or, is it feasible still for farmers to do this? Any perspective there from that whole agroecology integrating livestock perspective would be great.
Thanks. I think there's great momentum in thinking about livestock reintegration on cropland, especially in organic systems that rely on the soil ecosystems for production. So there's a real opportunity, especially in California, I would say, where we have a diversity of production systems and animals in the landscape that need to be integrated across various scales to provide forage over time, and weed control, pest suppression, etc. So I think things are changing, especially since livestock integration is also a pillar of a regenerative agricultural model. There's more and more talk about soil health, and we see in our results that there can be some benefits, even though they might be on the longer term. So it's a great fit for organic system in particular, and we see in our landscape actually more and more also conventional growers adopting sheep grazing in orchards and in vegetable cropping systems to deal with common production challenges. It remains quite knowledge intensive. It has associated costs to it. And the benefits will vary according to the structure of the operation and access to knowledge, extension, and relationships between shepherd, animal handlers, ranchers, and crop growers. So there's a lot of social economical components to bring those systems forward. But I hope that the research we're collectively doing today is starting those conversations and catalyzing more efforts across different regions of the US.
That's great. And so I guess highlighting that social and economic component, we would expect that those sorts of things would differ also in other regions. And so I'm curious from Emily and Fawzi how that looks in your different regions in Minnesota and Maryland, like the feasibility of livestock integration. Farm sizes can look very different in different states and regions, and the types of crops that people are growing. So like the feasibility of integrating livestock with all of these other considerations, how does that look to you all?
Actually, in Maryland, we have almost similar results to what has been reported in Minnesota and California. But the question I have is that these three regions, they have different climatic conditions. And the question is how we can apply this data to the rest of the country with diverse environmental factors. Because as we know, the main purpose of animal grazing is mainly soil health and its effect on food safety in that, yeah. So the question that needs to be discussed, how can these results be applied to the rest of the country, especially in the South, where they have different environmental conditions.
That's a great question.
So I think in Minnesota, some of the challenges would probably be more so on managing a good cover crop to be grazed. I think the willingness is there. I think many farmers want to include livestock. It also depends on whether they have access to livestock and how large their production is. I know that one of the challenges that we really found was establishment and water usage and the timing, because we have such a short season. It's just such a different animal. Even as we're trying to figure out how California's doing it or how Maryland's doing it, you know, we're all grazing at different times. And while California was planting, we had like 6 feet of snow. So it's just a very different growing season. We're just limited in that respect. And we also have such different soils. We have such high clay soils. So when it's wet and cold, it's wet and cold. And it's hard to terminate the cover crop to get the main crop in. And when the main crop is the moneymaker, you know, it's a little bit of a give and take. So I would say that it would be more of management issues would be a little bit tough, but not anything that I think is a huge barrier. And again, like Amelie said, it's a huge, you just have to go in. It's a huge learning curve. So that's right there. So more extension programs, things like that would be greatly helpful.
Mm-hmm. And Sequoia, since you were also, you know, knee deep in the field with the animals in California, even though the growing season is longer there in California, there are other challenges that could make this really difficult for people to adopt, or is it just easy because everything is so easy in California?
Yeah. Well, I can't imagine how you all got 2 grazing events in though, so we only managed to get 1. Because we were limited by water, really, and getting the cover crop to grow. Because if you have a dry year, there's just nothing grows in the winter. So that was what we had the first year, and then, yeah, just like Amelie said, kind of like in terms of implementing it between ranchers and growers, there's like that relationship building that has to happen. Like trust has to be built between people who want to try this out, and trying it out on a smaller scale is always important as well. And then there's also the component of predators. I don't know if this is if Minnesota has predator concerns, but we do have that. So ideally, if you're hiring out a contract ranger, you would have somebody who would bring like livestock guardian dogs who would keep the livestock safe, rather than me in a van sleeping over at the field keeping them safe, which is what we did. So yeah, but there's always those concerns about like timing of field operations, and ultimately farmers want to make sure they're seeding their cash crops at the appropriate time and not going to have concerns with like nutrient immobilization if they go too close to a cover crop or something like that.
Yeah, that's great. So I loved your question, Fozzie, about how, you know, how do the results that you all have relate to the rest of the country and all the other regions? How does it, can it? And I just keep thinking about all these different combinations of everything that you all had at the different field sites, different cover crop species, different crop species. We've had questions coming up actually from the audience about why you chose what you chose to work with. As far as cover crops or crop species, you know, the way that you managed this, was it when you're making these choices based on feasibility, on climate, on like methodology? So for instance, if you were to replicate an experiment in 3 different regions, one might expect that you would want to keep as much held the same as possible. But what you have presented today has shown that the weather is totally different, for one thing. But then also there are these socioeconomic factors too, like, are people interested in growing spinach in Maryland as they are in California? I don't know. So I don't know, I guess I'd love to hear a little bit more about that sampling design and how, maybe even just how it works to do such large scale multi-regional work when you're trying to get these results that can transfer across regions. Alda, maybe you could tackle that as the lead of this work.
So well, I also have to say we did this during COVID. So that was extra challenge for us, you know, as we were following different recommendations, and in California we're we're able to keep the research going on because it's a time sensitive. So that's extra challenge of these multi-institutional, and you know, much appreciate all the people and the team involved and the students that make that possible. So that was what we planned and what happened. So for example, in California, you see we have the same mix. First year we have a type of mix of cover crop. Second year we have a lot of daikon. So the sheep didn't like. So did that affect actually the behavior of what we see? We really not know. Within the same study design, just things turns out different because the cover crop didn't grow so much. There's a different, the sheep, they didn't like it that much. So it'll be interesting, you know, I'm imagining my next study design, it'll be interesting to do different cover crops, different species, right, in the same state, and see what that affects. So it's a challenge, but at least, so we try to absorb that differences by repeating the study across the states and control what we were able. For example, in the study design of the analysis that Sajin shows, it does matter the time, it does matter the conditions. Those are significant in our outcomes. So the different species, goats and sheep, again, they have different behaviors. They like different of the grass. One are more grazer than another one, so they might, you know, and how that affects, actually there's open questions towards that. Although in California we have consistent data over 4 years, and that's really, you know, it really tells something about the risks of these systems, as was consistent the findings over the 4 years. We didn't present the 4 years, but that was very much consistent of those 4 years in the same plot. So we control for the plot, control for the same sheep. We use the same sheep, so you have on farm sheep, or the same flock, I will say. So we try to control for all those, but it's a challenge, definitely. And that's, you know, these multi-state are so, I'll say they are a challenge, but make things much more interesting and applicable, I'll say, as you know, after 3 years working on this project.
Yeah, that's great. And I see Adrian has popped up, which tells me that we are at time. I'm sorry that if I didn't give some of you panelists the time that you were hoping to say your last words here, but we need to move along. So just thank you again so much for sharing all of your knowledge and expertise with us. And I'm going to hand it over to Adrian to do the last word.
Thank you so much. Thank you so much, Amber, and all of our other speakers today, Alda, Amelie, Emily, Sajin, Sequoia, and Fozzie. Thank you so much for your presentations today and for this great discussion. We really appreciate you not only taking the time to be with us today and share your research, but also overall for your commitment to organic research. Your work really does make a difference. So thank you all for the work that you're doing to move the research forward. I do just want to take 1 more minute here to highlight an in-person event that we have coming up in September in Philadelphia that the Organic Center is hosting. We don't always have in-person events where you can connect with us and learn more about the science behind organic, but we do have an event coming up at Natural Products Expo East in September in Philly, where we're going to be highlighting some specific projects from the Organic Center. We're going to have some art installations, and you'll have an opportunity to connect with our staff and learn more about the research. So if you're going to be there or can make it, please do join us. I also welcome you to check out our on-demand library of science education online. I just posted a link in the chat where you can access that on the Organic Center's website. We post all these online webinars within 2 business days, so you can learn about a wide range of organic science topics, such as recent webinars on organic spices, organic dairy, organic fiber, and a lot more. If you're interested in other potential research opportunities, reach out to Amber at any time. We are always happy to connect and hear your ideas too. And last but not least, thank you all for taking the time to join us today for the webinar from wherever you are in the world. We appreciate you taking the time to learn with us today. As a reminder, you are going to see a survey pop up on your screen in just a minute when you leave the webinar. Please do take a minute to complete that survey for us. We really appreciate it. And in the next 2 days, you're going to receive an email with a roundup of all of this awesome information you learned today. Don't hesitate to reach out if there's something else that you didn't get a chance to ask today. We hope that you'll all join us for a future organic science education webinar or other opportunity. And I hope you all have a wonderful rest of your day. See you next time.
Thank you.